So our first learning unit to start out Biochem 2 starts to get into some nitty-gritty of what biochemistry is really about, and that's learning how enzymes work. So we're going to build on a lot of what we talked about in Biochem 1, which is structures of biological molecules. And now we're going to learn how enzymes kind of do neat things with them. So in particular, in this chapter, we'll start out by just kind of remembering some mechanisms. We finally have a home for so many of those reaction mechanisms we learned last year. And we're going to learn how 3 hydrolysis reactions work. And so we're going to begin by reviewing what the hydrolysis reactions are that break the linkages between carbohydrates in glycosidic bonds, and RNA in phosphodiester bonds, and proteins in peptide or amide linkages. And so we'll do a little bit of a review there. We'll kind of review some of our amino acids and remember those important ones that have functional groups that allow us to perform chemistry. And then we'll take a deep dive into 3 enzymes in particular. We'll talk about lysozyme, which is found in tears and mucus secretions and is responsible for breaking glycosidic bonds. And even though it's not something that's involved in digestion, it's something that's really important as an antimicrobial agent for you because it helps to break down bacterial cell walls. So that's why you have a lot in your mucus secretions and in your eyes to kind of provide that sterile environment. RNase A, again breaking phosphodiester bonds. We'll take a deep dive into how that enzyme active site works, and then we'll make sure that we understand that that's why we have all of these RNases on our hands, and why we have to wear gloves if we're working with RNA in the lab. That whole idea of you are more hazardous to your science than your science is to you. And you have all of these RNases on your hand because RNA is not meant to be something that's more than a transient molecule in your body. You transcribe DNA into RNA, into messenger RNA, to make proteins, but those messenger RNAs are fairly short-lived. So your body knows that any RNA that's sort of long-term might be something that's problematic, and it might be a virus. So you make RNases to get rid of it. The last enzyme that we'll study really does have something to do with something that is digestion related, which are it's actually a class of enzymes called serine proteases. Excuse me, and serine proteases are responsible for breaking that peptide bond between many proteins that you might digest. So these are enzymes that are secreted in your digestive tract. They're produced in your pancreas, and so we'll spend a lot of time kind of learning about how those guys work. Big picture thing just to kind of highlight in this video, 3 things that we always need to think about when we talk about what makes enzymes work well, or how they work. The first thing is we need to do chemistry on our substrate. We need to use all of the reagents that we found in our uncatalyzed reactions. So those are our reaction mechanisms, our BRMs from last year. And then we need to make sure that we return our enzyme to its active state. And so all of the steps that we see when we study an enzyme active site have to do with those 3 steps. So we'll make sure that we understand where and when they happen, and then we'll actually have 3 role-playing activities that we utilize to act out all 3 of these enzyme active sites. So a fun chapter on enzyme catalysis.